Here we are. Hello, 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 hello. How are you? Kids, today I'm coming with one of the topics in unit 1 of grade 12, that is tissue culture. Tissue culture. Therefore, we shall briefly see about tissue culture as one part of biotechnology, of course, in plants, of course, specifically in plants. First, let us ask, what is tissue culture? Tissue is a group of tissue means a group of similar or identical cells communicating and joined together in a physiological way, communicating physiologically. Therefore, a group of cells functioning together, known as tissue. Therefore, tissue culture in this context is the growth, can be defined as a growth of tissues or cells in an artificial medium separate from the parent organism, plant or animal. Therefore, you take a sample of cells. You can take even a single cell or a group of cells or tissues, and can grow into an organ or the system or organism, the whole organism. Especially when you talk about plant tissue culture. Of course, plant cells are unique. I will talk about this unique in such a way that if you take any plant cell or any plant tissue and provide nutrition and the appropriate growth factors, growth regulators, or plant growth hormones, that sample cell or tissue can give rise to the whole plant. This is the unique ability of plants. This is different from animal tissue, of course. Therefore, tissue culture, simple definition in this context, tissue culture is the growth of tissues or cells in artificial medium by providing nutrition, nutrients, and appropriate condition plus growth regulators or hormones, growth hormones in an artificial medium separate from the parent organism or plant or animal. Then we have, if you say tissue culture involves both taking sample of tissue from plants, sample of cell from plants, both animals. We have plant tissue culture as well as animal tissue culture. But remember, in this context, in this grade 12 tissue culture part, it talks only about the first one, plant tissue culture, its application in agriculture, how much important significance we shall talk. Therefore, plant tissue culture and animal tissue culture, we are not going to talk about animal tissue culture in this case. Therefore, we focus on plant tissue culture. Now, plant tissue culture, as you observe, we define plant tissue culture is a technique of growing or producing whole plant from sample cell or sample tissue taken from a plant, parent plant, and growing on artificial nutrient media. As you observe here in this picture, you provide the sample cells either liquid media or solid media, even semi-solid media, which provides the appropriate nutrients for cell division, and then cells differentiate and give rise to the whole plant. Just take one single cell from a plant, any part of the plant. It could be the stem, the leaf, the flower, the root. Possible to grow into tissue, then the tissues grow, then give rise to the whole plant. This is one unique ability of plants. Therefore, how can you define plant tissue culture? A technique of growing or producing whole plant, the whole plant, just from sample cell or sample tissue taken from the parent plant on an artificial. You are growing on artificial medium in the lab. Therefore, we can say it is sort of plant cloning. When you say cloning, it refers to giving rise to the whole organism, to the whole animal, by taking sample cell, just a single cell, from a single cell growing the full animal. Of course, it's much sophisticated and very young field of biotechnology or field of genetics, cloning. When it comes to plant tissue culturing, just you can take any plant tissue from any part of the plant, so that provide nutrition and suitable condition in the lab, it will grow into the whole plant, so that you can propagate it into a field for crop production. Therefore, it's a very fast way of getting seedlings. Yeah. Then it is also known as micropropagation. Micropropagation is the other name for plant tissue culture. Then when it comes to plant tissue culture, is possible because of the unique ability of plant cells. You see, this is the secret behind plant cells. You know, plant cells are plants are a decentralized body. That means they don't have nervous system, and the whole plant body is a decentralized system. And because of this, every tissue or cell in the plant body has the ability to grow into the full organism. That ability is known as totipotency. Totipotency is the ability of every cell from every part of the plant, if provided nutrition and appropriate conditions plus growth factors, it will develop into a full plant. That is totipotency. This is not observed in the animal world, totipotency. The ability of any plant cell or tissue to develop into the whole plant is what you call totipotency. Because of this unique property known as totipotency, you can grow many seedlings, little plants, just by taking sample cells or tissues from a plant. That's why they can divide as young cells, differentiate, divide, differentiate, differentiate, and redifferentiate if they like, differentiate, de-differentiate, and redifferentiate continuously to give rise the whole plant, to give rise to the whole plant. This is unique when it is compared with animal tissue, animal cells. The process of plant tissue culture done as follows. Let us see the practices or the processes, the stepwise processes. First, excising or cutting a small sample tissue from leaf or stem or root, any part. Just you remove a small sample tissue from any part of the plant, and this sample tissue is known as explant. They call the sample. You can cut part of the leaf and prepare a group of cells, tissues for your tissue culturing. Therefore, that's known as explant. Explant can be taken from either the leaf, the stem, the root, any part if you like. Then place it in a sterile nutrient media at suitable temperature, totally sterile, free from microorganisms, contamination. We should not contaminate the media. The media or medium, singular medium, is a surface in which nutrients are applied, nutrients for cell division, plus that contains appropriate plant growth regulator or plant hormones. Therefore, medium, you provide sterile nutrient medium, either in a bottle or petri dish. You provide this for the cells, the sample cells. Therefore, you put the sample, place the sample tissue in a sterile nutrient media or at suitable temperature. Then the explant divides mitotically, mitosis, to give rise to a regular, unorganized, and undifferentiated mass of cells known as callus. They call it callus. If you can cut this callus into many parts so that each cut piece will develop into another big mass, and each big mass will develop into seedling if it is given appropriate growth factor, growth hormone, plant growth regulator if given. Then we put the sample tissue in a sterile nutrient media at suitable temperature for cell division to be fast. Then the explant divides mitotically to give rise to what do you call that? Irregular, unorganized, and undifferentiated mass of cells, that is callus. Then the callus is further divided into smaller pieces, and each piece is put in a new nutrient medium, and each piece again grows bigger. Cell division. Now cell division. Therefore, we give plant growth regulators which facilitate differentiation, cell differentiation. Then appropriate plant growth regulators are added to stimulate cell elongation and cell differentiation. Then cell division, cell elongation, cell differentiation. What will happen? Finally, the masses of tissues each give rise to plantlet. Plantlet means small plants that are transplanted. Of course, these are small plants. ችግኝ ማለት ነው። ችግኝ ማግኘት ችግኝ that are transplanted onto field soil or in greenhouses. Greenhouse. You see, very fast and economically also feasible. Just no need of sowing your seed. Of course, this is asexual. This is asexual. You take sample cell from a plant and give rise exact copy of that plant. This is asexual, perfectly asexual, artificial asexual reproduction, because it is done by humans in the lab, providing suitable temperature, suitable nutrient media, and taking the sample, putting so that you control it, you bring about the formation of plantlets. Plantlets are small plants. Then the plants resulting from each technique are all identical, as they are all derived from a single parent plant, and they are, of course, they are known as soma clones. Clone means just soma clones are those plantlets obtained from that sample tissue. They are all the same because they are asexual. Plant tissue culture is asexual. Sexual propagation, micropropagation is asexual, done by human hand. That is artificial asexual reproduction. Therefore, these plantlets obtained from sample tissue are known as all soma clones. You can see diagrammatically here. And now we are using leaf as an organ to give us sample tissue. We take sample tissue from leaf. Sample tissue or cell called explant is scrapped from any part of plant, in this case the leaf. The sample plant tissue or explant is placed in nutrient medium and plant hormones added. The sample tissue divides to form a mass of undifferentiated cell. We have called it callus. Over time, each fragment of tissue, we fragment it, then the callus is divided and put in a new nutrient medium with hormones that facilitate cell division, differentiation, elongation. Then each fragment of tissue then grows into a small plant known as plantlet. The plantlets are then planted in a plot of land or in a greenhouse or a field, usually in greenhouse. By the way, since all plants obtained by this plant tissue culture are all identical, we have to care about sanitation. If one plant is contaminated or affected by virus or bacteria, disease, and if that disease is transmitted throughout the soma clone, throughout the group of plantlets obtained from the same tissue, since they all have the same genetic makeup, it will destroy all 100%. That means it's a big economic loss. Therefore, we take care of their surrounding to prevent contamination, not to reach to these plants. Then, this tissue culture technique is a very useful agricultural technique, widely used in horticulture and floriculture. Horticulture involves also fruticulture. Horticulture, cultivation of vegetables, fruit plants plus flower. Floriculture is flower production. Of course, horticulture is much broader sector of agriculture and floriculture. Therefore, this technique, plant tissue culture, is widely used in horticulture and floriculture. It is also extensively employed in large-scale production of tomato, carrots, plant trees, palm tree, orchard, banana, apple. All these plants have been propagated or multiplied for their products by using plant tissue culture. Now let us see what's the advantage. What advantage, agricultural advantage, does this plant tissue culture has? One, to get a large number of seedlings from small sample of tissue within short period of time. Time efficient plus economically. Just you take sample tissue, you provide nutrition and appropriate plant growth regulator so that you will get large number of seedlings just from small sample of tissues taken from the parent plant. This is one advantage. Short period of time. The second, healthy plants can be grown from diseased plants. How? This is very interesting. I will come to that. How can we grow healthy plants from diseased plants? If we have a diseased plant affected by virus, bacteria, or any fungus, plants do have special groups of cells as their tips, shoot tip or root tip. These special types of cells are known as meristems. I will come to this point in a much wider aspect. Meristems, and these meristems lack vascular tissue so that the virus or the bacteria or the fungus will not reach to the meristematic cells. So when you undergo tissue culture, if you take from the diseased plant, if you take meristematic cell samples, these meristematic cell samples, since they don't have any virus, bacteria, or fungi, will give rise to a healthy plant. Healthy plant that is exactly the same to the diseased plant. Therefore, the way we produce healthy plants from diseased plant is just by using meristem culture. Meristematic cells are taken, not cells from the leaf, not the stem, just at the tips, shoot tip and root tip. There are special regions. I'll show you. I do have a diagram here down picture. The other advantage is plants without seeds can be multiplied, like plants without seeds, like banana. You can multiply banana by using tissue culture because you don't have seeds. The fourth advantage, useful. This plant tissue culture is useful where sexual reproduction is impossible, like in case plants with no seeds are there. It can also be used to propagate high-yielding disease-resistant varieties in a short period of time. High-yielding, disease-resistant varieties of plants within a short period of time. You can propagate, multiply within a short period of time. That's this advantage. It is also used to recover health plants. This is a repetition, of course. I have tried to tell you about meristem tissue culture. It is also used to recover healthy plants from diseased plants by taking only the meristematic cells. Let me show you here. Diagram I do have. This diagram tells you the first one is, you see, from the tip, meristem. It says you take meristem from the shoot tip. This is the region where meristematic cells are found. These meristematic cells will not receive virus, bacteria, or fungi if the plant is infected by these microbes. Will not come to here. Therefore, this region is sterile, totally free from microorganisms, pathogens. So if you take sample cells, there is no virus, there is no fungi or bacteria here. Therefore, you can use these meristematic cells as sample cells and grow healthy plant without disease. Therefore, this is growing disease-free plant from diseased plant, or a plant affected by pathogens. Therefore, by taking this type of culturing, plant tissue culture is known as meristem tissue culture. The other is, of course, you can take from the stem, you can take from the leaf, even you can take from the root, and sample cells are taken from the leaf, from the stem, from the root, and grown to give rise to the whole plant. You can't grow disease-free plant by taking sample tissue from the leaf or from the stem or from the root, because in this root, the stem, the leaf, the pathogens are found there. Vascular tissue is there. Vascular tissue brings the microbes, the disease-causing pathogens to this area, so that you can grow disease-free seedlings from this part, the leaf system, and you have to go to the meristem. In this case, the shoot meristem. The meristems are totally free from microbes. This is the essence of meristematic tissue culture. Then, and this is because the meristematic cells are always free from the pathogens and they lack vascular supply. Vascular supply means xylem phloem, they lack. Substances come here just by diffusion. Diffusion will not bring the virus, the bacteria, and the fungi. This process of using meristems for tissue culture is known as meristem culture. And therefore, if you are asked what is the significance of meristem culture? Oh, to grow disease-free plants from plants affected by disease caused by virus, bacteria or fungi.